Good morning, everyone. Let's start with taking up a few questions from the study guide, and then we're going to wrap up lecture two and start lecture three today. So here is a pedigree question. I forgot which one it is, but it's in the study guide, and it's asking us to figure out the possible blood type for question mark right here. So let's take a look at people whose blood type we know for certain about the genotype. That would be type O. So type O, there are no questions that type O will be little I, little I. Okay? Same thing for people with type AB blood, right? We know they're going to be IA, IB. Right? As for everyone else, we can only fill in half the information initially. Okay. The missing half, we can try to use logic to figure out. Um, Sometimes it's possible, sometimes it's not possible. We'll see. So that's that. Now, these two people have two daughters, and one of the daughters is type O, which means one of the little I came from one parent, and the other little I came from the other parent. Okay, so it's always good to look at people related to someone who's type O, and that will give you idea, of what the previous generation might be or the next generation. Okay, how about over here? Well, this person is type B, so there are two possibilities for his blood type. It could be IB or it could be IB little I, right? But because his daughter is type A, it's impossible for him to be IBIB. If he was IBIB, then the only thing he can pass on would be a IB, which would make the daughter type AB blood. And since she's not type AB, then that means the father has to be IB little I, and the little I is what got passed on to the daughter. Just gonna let you think about that for a second. Okay. So what about the IA? Where does the IA come from? Well, IA is going to be from the mother. Okay. And where did the mother get her IA from? Well, if you track it back another generation, she must have got it from her father. So what about the missing half? Well, the missing half is going to be from her mother. And right now, we do not have enough information to figure out whether she got the IB or the little I. Right? Like they would have to have more kids for us to figure it out, right? So the correct answer, it says list all possible blood type, right? It will be IAIB or IA small I. Okay. So on the test, the question might be, I give you this pedigree or some other pedigree, and I would say, select all possible blood type for whoever, right? Then it would be like a multi-select question, right? Any questions?
I have a question, sir.
Mm-hmm.
Yeah.
Yeah,
Just
go
to
ahead.
just to understand blood type
Yeah.
for me I thought it was only to determine if it was A or B or AB, but not to the
Sorry,
genotype.
the genotype, did you say?
Yes. This is the genotype of
Yeah,
the
this is genotype, yeah. Genotype, right.
yeah. I said the question I didn't understood it like you gave the genotype. For me it was the phenotype only.
Oh, like you mean the blood type? Is that yeah?
Yes.
Oh yeah,
Yeah.
okay, sure. Yeah, yeah. So this is genotype. So therefore her blood type is gonna be type AB or type A, right? Is
Okay.
that what you mean? Yeah.
Yes. Yes.
Okay. Yeah, thank you.
So this kind of question we have to answer that like this.
Yeah,
Just
could be. Yeah. So it would be a multiple choice question, and if I ask for blood type, then yes, I would definitely give you blood type. If I ask you for genotype, then I would definitely give you genotype. Okay.
okay. And the last question. Yeah. The for the A, the last daughter.
This
How?
one.
Yes. How
Mhm.
you say that is only I that it can give, not
How do you know it's little I here?
yeah, literal I.
Sure. So, okay, let's backtrack a little bit, right? The father is type B blood. So what are the possible genotype for someone who's type B?
Capital B, little I.
Okay,
Um,
IB little I or what? How else can someone have type B blood? What other genotype is gonna give him type B blood? Other people feel free to jump in if you want. Yeah, that's right, Cheyenne. IB, right? Do you agree, Elvis?
yes,
Yes, both of these would give him type B blood, right?
right,
However, because the daughter is type A, it's impossible for the father to be IB. If the father only has IB, then the daughter will guarantee to inherit an IB allele, right?
right.
And that means she would not be type A blood. Right?
Okay,
If you get an IB allele, you're gonna have a B, right? Whether you are like AB or B, it doesn't matter. There's gonna be a B. But the fact that you have an A type blood daughter, that means the father must be IB little I, and
okay.
and he has to pass on the small I. Is that better?
Yes, yes. Thank you.
Okay, no problem. Anyone else have any other question? All right. Let's do one more matching question, and then hopefully we have enough people showing up because we're gonna take a big note um, right off the bat. Okay, so this is another one of those baby questions where you have to match them up. Um, you will have a matching question on the test, so please familiarize yourself with it. Okay, so what's going on here? We have a bunch of parents, and we're trying to match the babies. Um, Sorry, I gotta restart. This program has been like really glitchy lately. There we go. So, let's take a look at the first couple. Okay, so the mother is AB, and then the father is O. If you are AB, you cannot have O babies. If you are type O, you cannot have AB babies. These are the rules we wrote down last class, right? Over here, right? There you go. Put them on a sticky tag, stick them on your wall. And Kendra is telling us that it is baby H. Well, how do we know that? Both of them are type negative. That means it's impossible to have positive baby. So baby H is theirs. Okay, so that's gone. Next, we have A positive and O positive. O positive. So type O cannot have AB baby. So that's not theirs. Uh, but I don't have enough information at this point to know whether it's F or G. It's one of them. So I'm gonna put down both for now, and then we will see if we can eliminate one of the choices. Okay, but definitely not the first one, right? Because the father is type O. Type O cannot have AB. Next, we have another type O mother, so not baby E. Um, do we know whether it's F or G in this case for the third couple? What do you guys think?
Yes,
Yeah, which one is it?
is B plus. So
Okay, some people will say F, some people say G. Why don't we do a vote? Okay, let's see what you guys think. All right, let me do a poll. Okay, choose A if you think it's this baby. Choose B if you think it's that baby, and choose C if you think not enough information. Okay, we're
Sorry,
talking.
which
Hmm?
baby are we solving for right now?
We're trying to solve for couple number three. Do
Oh,
Well, we know for certain it's not the first baby, but do we know whether it's baby F or baby G? Choose A for baby F, B for baby G, C for not sure. Hey, so I have some good answers coming in. And yes, the correct answer is baby F. It cannot be G because G is type A, and neither of the parents can contribute the A allele, right? So you cannot have a type A baby. So for that reason, it's going to be F. And since F is the only choice for couple number three, it wouldn't be couple number two's baby. So by process of elimination, we know G is for the second couple, and the only one that's left would be E for the fourth couple. Now, some people get confused because they see two positive parents and then they see the child is negative, and they are not sure how that's possible. Remember, if you're positive, you can be heterozygous positive, right? So positive, negative, positive, negative. If you do the Punnett square, you see there's a 25% chance the baby, would be Rh negative, right? So if both are negative, then all the kids are going to be negative. However, if both are positive, it's not always necessarily going to be positive for all the kids. I mean, it would be if both of them are homozygous, right? If both of them are positive, positive, like if the parents are positive, positive, and positive, positive, then yes, it's going to be 100% Rh positive, right? But if they're heterozygous, then there's 25% chance that the child will be negative. Are we okay? All right, so that's the end of blood for the most part. Now, I'm going to give you a note, and it's what should we call it? We'll say introduction to lymphatic system. It's gonna look familiar to some of the stuff you already know at first, but copy it anyways, okay? Because I'm gonna add more detail to it. It will be a good review anyways. All right, so I suggest you do your paper sideway. Take out a brand new sheet, okay? We're gonna draw some stuff here, and then we're gonna draw some stuff here, and then we're gonna, loop back to the right side. All right, so plan your space accordingly. Start on the top left, and we'll come down here, and then we're gonna end up over here, and then we'll write some more stuff on the side, okay? This should take around 15, 20 minutes. All right. What do we know so far? Well, we know if we have a blood vessel like this, If we have a capillary, we know capillaries are leaky. Okay, so I'm going to put some gap there. Of course, this is an exaggeration. It's not actually that big of a gap, but it's leaky. Okay, so this over here is a capillary. Okay, make this big, all right? Lots of detail to put on it. We also know one side of the capillary is connected to the artery. So we call this the arterial side. And on the other end, it's connected to a venule. So we call this the venous side. Okay. Now, inside the blood vessel is going to be blood, and the blood is not separated. But I just want to make a point to help you remember some of these things, okay? What is blood, anyways? Well, we learned that blood is mostly water and cells, and you can separate it by spinning it down. And so this yellow stuff that I've drawn here, that would be like the plasma portion of the blood, and it makes up approximately 55% of your blood volume. Okay, it's mostly water. It has some electrolytes, some waste products, right? And proteins, lots of proteins in it. That's your plasma. Okay. Now within this plasma, um, the cells would be there. So we have these red blood cells, and we just got to draw a few there. And red blood cells, they are the hematocrit of the blood, and it makes up a majority of your blood cells. You have a little bit of white cells. Okay, maybe a white cell here. Okay, remember white cells, they have multiple nuclei. There might be another white cell here, but they are not very abundant, right? Like you have a lot more red cells compared to the white cell. Okay, so these are RBC, and then you might have some WBC. And let's not forget about our platelets. Okay, they're really, really small, just swimming around. They're not really cells, they're just like cell fragments. Right? So of course everything is all mixed together. You're not going to be able to see the plasma, separate from the rest of the blood component. You can only see the plasma if you spin down the blood, right? But I just want to put it here for you to um, to show you, all right, to remind you. So far, nothing is new. And we also know that because of blood pressure, there will be an outward force pushing out fluid, pushing fluid out of the capillaries. Okay. And we drew it all the way, but let's not do that. Let's just draw two arrows here, beginning and end. You know it's there throughout the whole thing. And I told you blood pressure on the arterial side is approximately 30 millimeter mercury. And as you move further and further away from the arteries, the blood pressure will continue to drop, and then it's about 15 mmHg millimeter mercury on the venous side. You will know this. You did a quiz already, right? You know blood pressure is going to drop as you move away from the heart. What's the other force? What's the name of the opposing force that's going to be drawing fluid back into the blood vessel. OP, that's right. OP. I think I used purple last time. Let's use purple again. So OP, which stands for osmotic pressure, is going to be pulling fluid back into the blood vessel. And that's caused by a difference in concentration of solute on the inside compared to the outside, right? So OP here, again, this is nothing new. You know this already. Okay, I just need to set the stage to introduce the lymphatic system. OP is about 20 mmHg, roughly. Okay, about 20 mmHg. And so on the arterial side, we have a net pressure that's uh, pushing fluid out. Over here, we have a net pressure of drawing fluid in. Okay? So fluid is going to come out. Again, not blood, it's just the fluid portion. It's mostly water. It might have some dissolved electrolytes in it. All right. No proteins, no cells, they're too big. And it's going to come out. And what's out here? Okay, we did not draw this last time, so I want you to draw it now. Okay, we're just gonna draw a bunch of cells. Okay, they are like sunny side up eggs. Okay, take a moment, draw your best looking cells. Okay, that's the nucleus in the middle. Okay. Now some cells are more closely packed together than others. They don't always have a gap, but don't worry about it for now. Just draw some cells. Go. There we go. Nice bunch of cells. Okay, so these are cells. Cell in the tissue. Okay, I'm just going to give you a moment to catch up. So all these fluid that's leaking out, it's basically going to um kind of go into the space between the cells. Okay, and I hope you are using colors. It's going to basically fill up the space between the cells. A highlighter probably works better in this case. There you go. Now take a look at this. The fluid that's coming out, and it's coming out because again the BP is greater than the OP, right? So the fluid's gonna be basically filling up the space between the cells, and some of them is gonna make it back to the other side of the capillary, and and they're gonna return to the blood vessel, right? Because on the other side, keep on telling you, blood pressure is now lower than OP. And so the osmotic pressure is drawing fluid back in. So we have a name for this stuff. Okay, the fluid between the cells. Where should I put it? Okay, I'm just gonna put an arrow here. You put it anywhere you like. Just make sure you're pointing to it. These are called interstitial fluid. Okay. They are between cells in tissue. Okay. Interstitial fluid. Is it clear so far? What's going on? No, no questions. Okay. But we have a problem. If this is the only thing that happens, then we have a problem. And the problem is we are losing more fluid from the capillaries than we are able to bring back in, right? Look at the pressure difference. Here is 10 and here's only 5, right? So you have more things being pushed out on the arterial side than you are able to bring back in. Sure, you can bring back some, but over time, the fluid is gonna basically overflow, right? You're gonna have too much interstitial fluid, and if you don't drain these fluid, they will start to build up in the tissue, and then it causes it to swell up, and you will have edema, right? Edema, edema, right? Swelling. So we don't want that, and that is where the lymphatic system is going to come in, right? Next to your capillaries, you will always have what we call a lymphatic vessel that is nearby. Okay, so this is a lymphatic vessel. Okay, always near capillaries. Lymphatic vessel. And the purpose of the lymphatic vessel is basically provide drainage. So these excess interstitial fluid that didn't make it back to the blood, they will enter the lymphatic vessel. And as soon as they enter the lymphatic vessel, their name changes. Okay. Once they're in the lymphatic vessel, I'm going to change the color to make it green. They always use green for a lymphatic vessel. I don't know why. They are not actually green, obviously. Um, as soon as they go into the lymphatic vessel, now we call it lymph. Lymph. Okay. Which is fluid inside lymphatic vessel. So it's gonna flow. Now, there are no pump connected to the lymphatic network. Okay. The blood vessels, it's connected to the heart, which keeps things circulating, but we don't have any pumps in the lymphatic vessels. So um, it's hard to keep things moving, and we don't want it to pool at any particular location. So within the lymphatic vessels, you're gonna have one-way valves. Okay, these are valves. Valves. Prevent. Right, there's another valve there. Valves prevent um, fluid, prevent lymph. That's what they're called, lymph from building up. Okay. Hey, give me a signal when you're done copying, okay? So I can move on. I still have to add more. Just like, give me a thumbs up or something like that whenever you're ready. Thank you. Okay, I'm gonna move on, all right? Stop me if you need me to stop. Otherwise, I'm gonna keep going. So I hope you still have space, okay? Because we're gonna so far it looks kind of nice, but we're gonna have to draw more things to kind of ruin it a little bit, okay? So this is gonna keep going. Okay. And somewhere else in the body, we're gonna have another lymphatic vessel. Okay. And then they're gonna come together. Okay. And somewhere else in the body, we're gonna have another one. Okay. And maybe another one, coming together like that. Okay. Coming together, just like all over the body, collecting these excess interstitial fluid, right? And kind of draining it. And they will all come together like that. And then every now and then they would converge into a big ball-like structures. Okay, actually it's like a cluster of them usually, but I'm just gonna draw one big one. And then it's gonna go back to, and we're gonna extend our blood vessel here, like that. Okay. It's gonna go back to the blood vessel like that. Okay, so like eventually the capillaries is gonna become venules, and then it's gonna become like the vena cava, right? All all blood vessels eventually will come together and join with either superior or inferior vena cava before returning to the heart, right? So all the lymph will come together to this place, and then ultimately it's gonna return to the blood. Anybody know what this thing is? Yeah, I'll show you where some of it is, okay? And then maybe you can tell me the name. Okay, like you have some here, okay? You have some like under the armpit, right? In the legs and the groins. Yes, Cheyenne, excellent. Okay, 100% for you. This is the lymph node. The lymph node. Lymph node. Okay? Inside the lymph node, you're gonna have a bunch of white blood cells. Okay? You're gonna have a bunch of white blood cells. Okay, white blood cells. Okay, so these are white blood cells. And what they're trying to do is they are going to screen the lymph and check for pathogens. Okay? It's like a security checkpoint, making sure everything is okay. There is nothing dangerous here in the lymph. You may pass, and then it will go back to the blood vessels. Okay, so this is actually part of a surveillance um, mechanism. Okay, by letting the fluid kind of wash through the area between the cells, it will um, eventually go to the checking station in lymph node, and you'll be able to see if anything's wrong with the tissue. Okay? Do not do this next part. Okay, I'm just gonna show you. You can just watch. Okay, so look, let's say you have a thumbtack, jab into your hand or something, nail, and then what happens is you will start to have these bacteria in your system introduced from the nail, right? So the bacteria might be producing some kind of bacterial protein and antigen and whatnot. Those things will now enter your lymph, right? Because the fluid keep on washing the space between the cells, right? And it will carry some of these things into the lymph, and then it would get carried to the closest lymph node, And then the white cells would sound the alarm and say, hey, we have pathogen in the system. Hey, we have bacteria in the system. And then they will divide and divide. And then that's when when you have an infection, you can feel your lymph node swollen, right? The swollen lymph node is basically the white cells dividing and try to mount an immune response to help clear out these things. Okay, so the lymphatic system, yes, it's for drainage of excess fluid, but it's also there for surveillance. Okay, and if you detect something that's not supposed to be there in your body, you're gonna activate the immune system, which is lecture three. But is everything coming together for you, right? This is very exciting stuff, right? To see how the different system interconnects. Any questions for me? No? Well, I hope you have a lovely diagram that you that you understand. Um, I'm gonna put a little summary flow chart here for you in case you are still a little bit confused. So we have blood, okay? Because of the blood pressure, the fluid is gonna get pushed out of the blood, and once it's in the tissue, it's called interstitial fluid. Okay, most of it is gonna go back to the blood directly on the venual side, right? On the venous side, like over here. They come out, they come out on the left side, and then they just go back to the blood right away. Okay, but you're not gonna be able to pick up everything. So whatever you don't pick up is gonna go to the lymphatic vessels, and it's gonna become lymph. Okay? And eventually, the lymph is gonna go back to the blood. Okay, through the vena cava. Okay? And over here is just through venous side of the capillary. Right. So ultimately, you're not. If everything's working the way it should, then everything would ultimately, uh, everything that originated from the blood will ultimately end up at the blood. And when fluid is not supposed to be accumulating anywhere in the body, right? That is that. Let's take a look at our lovely PowerPoint slides. This is the lymphatic system. We're going to label um, some of these things in a bit. We already took the note. And this is lymphedema. Lymphedema. It's also called elephanitis in the local community where this is prevalent. As you can see, it looks very painful, and it is. And it is caused by blockage of the lymphatic vessels. Okay, the blockage can be there are various reasons. It could be because of like parasitic infection. It could be because the valves are not a damage in the lymphatic vessels. And it is, very, very painful depending on where it happens, right? And oftentimes for communities for places where this kind of condition is prevalent, um, they actually don't have access to the treatment they need to fix the problem, and so they have to live with it, right? And you can see this is very debilitating condition. But in North America, if you are diagnosed with this, then there are ways to fix it. First of all, of course, you would drain it to get rid of the excess buildup of the fluid. And also, they would have to identify the cause of the problem, right? If it's caused by parasitic infection, then obviously they need to, get rid of the parasites, right? If it's caused by like damage vessels, then sometimes they have to put a shunt in. A shunt is basically going to help you drain the fluid, reroute the drainage system in your body, for example. Okay, but that is lymphedema. That's why we need to have a functioning lymphatic system. Right? Otherwise, the fluid is just going to keep on building up and building up. It's like a overflowing uh, uh, what do you call those things? Like for the rainwater to go through, if that thing is like, overflowing cloth, then everything just back up, a sewage back up. That's what I'm trying to say. Anyways, that's that. Okay, lymph nodes, they are all over the body. Okay. This is the common one. And if you have like strep throat or anything like that, yes, your throat hurts, but then you can feel all the swollen nodes as well. Okay. And depending on other type of infection in the body, like the lymph node might be swollen in different areas as well. Okay. Um, we have other lymphatic organs to help you screen various things in the body. The tonsils is another lymphatic. It's not an organ. It's more like t lymphatic tissue, right? Organ would be like the whole thing, such as like like a kidney or something. That would be an organ. But tissue is just a mass of a collection of cells, and we have three pairs of tonsils. So this is your nasal cavity. Yeah, it's actually quite big in there. Uh, I'm sure, if not all, most of you would have had some kind of a COVID swab done on you. So when you stick the Q-tip in, right, like that's where you are swapping, okay? But further back, you're not gonna be able to reach that, okay? Like it's gonna hurt. But if you go further back, that's where you will have your adenoid tonsil. Okay, so everything that you kind of breathe in, the adenoid tonsil will be able to kind of screen some of those things. Like, are you breathing in, pathogens, right? And if you have a cold, uh, that's where the virus is going to infect, right? And then, it gives you stuffy nose and all those unpleasant symptoms. Okay, so adenoid tonsils. You have another one right here. This is called the palatine tonsil. You can see this in the mirror. If you look in the mirror and you say shine your phone at it, right? There you go. That's the palatine tonsils, right? Sometimes if you get really bad infection, you might see them turn orangey color, okay? Even white dots on it sometimes, okay? And that is a sign that you have some kind of serious throat infection most of the time. Yeah, yes, people can remove their tonsil, adenoid tonsil. There are sometimes they have these rocks that grows on them. It's not really rocks. They call it rocks, but it's like calcium deposit, okay, that grows on them, and it irritates the throat when they swallow and stuff like that. And so they might have to remove the tonsils, and that's okay. You can live without the tonsil. You might be more susceptible to getting infections, but it's not like a vital thing in the body. Okay. Uh, there's a uvula here. We'll learn that in the digestive system. But there you go. Okay, go check out your tonsils after class. Um, when the doctors stick the popsicle stick, right, the tongue depressor, which is really just a popsicle stick, it's press your tongue down, right? They're trying to look at the another pair of tonsils right there. Okay, that's called a lingual. Lingual is tongue. Okay, lingual tonsil. So three pairs of it: palatine, adenoid, and lingual. Right? So these things would be screening for, sorry, these would be screening for things that you are ingesting, and then this would be screening for things you're inhaling, basically. Okay. The spleen. The spleen is on your left side behind the stomach. You only have one spleen. It's not on both sides. And the spleen is the largest lymphatic organ, and its function is very similar to other lymphatic tissue and lymphatic organs. It's like screening for pathogens that is in the blood in this case. And it also acts as a reservoir for blood. Okay, it's able to store a little bit of blood. If your blood pressure is going down, it's able to, kind of provide that extra bit of blood for you, to kind of keep your blood pressure high until, equilibrium is restored. You can live without your spleen. There are various reasons for doing a splenectomy. And the person again might be more susceptible to infection. Okay. All right, next one. Thymus. Okay. Please do not confuse your thyroid with your thymus. Okay, thymus is here, right? Kind of a little bit above your heart. Your thyroid is up here, right? So the thymus is where a type of immune cells called T cells, which we will learn in lecture three, they will mature in the thymus. Okay, but only about 5% of the cells are going to be able to leave the thymus. They have to take a test. It's called a tolerance test. Now, you have to make sure these cells do not attack your own cells. Okay? If the T cells in the thymus recognizes your own antigens, then we have to kill them. We cannot let them come out. Yeah. And sometimes the body makes a mistake. We let them out anyways, and that causes autoimmune diseases. Okay. If you don't know what that is, again, we'll learn about it in lecture three. The gland does produce a hormone called thymosin, which will help with the maturation process. And that is it for lecture two. Okay, that is it for lecture two. Now, before we move on to number three, I do want to take a moment to label this thing with you. Um, I mean, I hope you printed it out. Uh, like I said, starting especially the second unit, we have a lot of labelings to do, and you really need to have these diagrams with you in class to make the most out of the learning experience. Okay, it's like you can't just watch me color it and you not do it. It's you really need to have these sheets with you. Okay, so this is part of the study guide, and so let's try to label some of these things. Okay, so up here in the head we have I guess it's kind of hard to see what they're pointing at, but they are pointing at the tonsils. Okay, so up the nose at the back we have the adenoid tonsil. Okay. And then this one, they are trying to point to, even I don't really know. It's either the palatine tonsil or they're trying to point to the lingual tonsil. It's one of those. I'm not going to ask you which one it is, so I'm just going to put tonsil. Okay, how about this right here? What is this structure? This organ right here, right? They took it out and make it bigger, but what is it? We literally just learned about it. Anybody remember? Yep, it is the thymus. Very good. For T cell maturation. Okay, let me put the thymus here. Okay, T cell maturation. Have I told you about T cells? I think I did, right? Like briefly, right? Like when we did the granulocytes and the agranulocytes. T cells, they help coordinate immune response. But don't worry, we're gonna have a whole lesson on T cells. Okay, let's skip some of these things for now. How about this little guy right here on the side? What is this thing? just behind the stomach on the left side. Yep, that would be the spleen. Okay, let's move down a little bit. Um, this here, this is bone marrow. Okay. Site of, I don't know if you remember this word, hematopoiesis. We learned this earlier in the lecture, which means is production of blood cells. Right? So T cell is a white cell. There are other kinds of white cells which are part of the immune system. They have to come from somewhere. Bone marrow, that's where you make them. Okay. They're missing a thing here, but the appendix sometimes plays a role in immune system in the lymphatic system. Never mind, don't worry about it. Okay, what else do we have? So here we have these little cluster of little grape-like structures. Okay, so those are the lymph node. Okay, that's just the thymus. Right? We already labeled it. This is another lymph node. Okay, we're gonna look at the lymph node in a bit. This is pointing at a lymphatic vessel. Right? It's all over the body. It's basically anywhere you have a capillary nearby, you will have a lymphatic vessel. Now this is a very, very busy diagram. I'm going to zoom in a little bit and I'll try my best to make it not confusing. Let's start with this one here. This one's not bad. Okay. The fact I'm using green, you probably already know what kind of vessel it is. Okay, so this would be lymphatic vessel. Okay. Lymphatic vessel also has like big and small ones. Sometimes the one next to the capillaries, they're so small, people call it lymphatic capillary. Doesn't matter. We just say lymphatic vessel. What's this thing right here inside the lymphatic vessel? Yes, the valve. We want to keep things going. It doesn't have a pump, so it doesn't move very fast. So we need to make sure they don't pool. Anytime the fluid goes through the a valve, it's not going to be able to come back. Right? So it makes sure things are moving in a one-way direction. Okay, over here we have, and I'm not going to outline all of them, but these are tissue cells. And then we have another arrow here that's pointing at the fluid between the cells. Again, if you're looking for a soothing activity to do later on in the evening, you can color the whole thing, but we don't have time right now. So this here is the interstitial fluid. Almost there. Okay. That would be the capillaries. Okay. Blood capillaries. Right? Like the blood vessels. Okay. And that's it. That's it. We know the fluid's going to come out from the capillary, okay, and then it's going to go into the lymphatic vessel. That is all for lecture two. All right, lots of stuff. All right, the genetics problem, different kind of white cells and everything. All right. How about we take an early break before we start lecture three? I'll leave this up for you if you need to, write down more things. Let's come back at 9:35. Okay, and when we are back, we'll do lecture three. So if you need to get those notes, please do so. Oh, there is a question. Why the nodes are just called lymph nodes instead of, say, lymphatic nodes? Uh, I don't know. They call lymph nodes, right? Not just nodes. We don't say nodes, we say lymph nodes, right? So lymph node, lymphatic node, same thing. Could you please scroll up to the diagram we drew? Yeah, sure, sure. This one? All right. So, 9:35. See you guys in a bit. Okay, let's continue with lecture three. So lecture three is about the immune system. And there are a lot of moving parts in this. So it could be a little bit confusing at times. We will, again, take a lot of notes for this unit. You can see there's not a lot of slides here, but we will definitely be writing a few side notes for you to understand some of the harder concepts. So here we go. What is immunity? Immunity is basically your body's ability to protect itself against incoming pathogens. Okay, so pathogens are, we talked about it before, they are living things that cause you to be sick. Right? So, bacteria, virus, fungus, parasites, all these things are pathogens. And they are always in our environment. Even if you, just pick up something and eat it, you are introducing pathogens into your mouth. Not all of them are gonna make you sick necessarily, because your immune system is always working, right, to get rid of them as quickly as they can. But in situations where your immune system fails to get rid of them fast enough, and then they, establish kind of like a foothold in your body and start multiplying, then it's gonna make you sick. When it comes to immunity, there are two key um, branches. Okay? We have the innate immunity, which are immunity that you are born with. Okay? The moment the baby comes into this world, it's already equipped with a bunch of protective mechanisms. We call these innate immunity. They are not really tailored to fighting a specific kind of infection. They're just like general defense mechanisms, okay? And they don't change throughout a person's lifetime. We're going to learn about these innate immunity today. But for example, like fever, right? Fever is an innate immunity. Right? A baby is able to have a fever when it's catch some kind of infection. An older person will also be able to have a fever. It's the same kind of fever. It's not something that would change throughout the person's life. Okay? Adaptive, which is also known as acquired immunity, these you get throughout your lifetime and it evolves over your lifetime. Right? Every time you get infected or exposed to some kind of pathogens, your body learn to fight them and you create protection against that. Okay. So your adaptive immunity is not necessarily same as mine because the kind of pathogens I got exposed to throughout my life may be different from yours. Okay. I mean, there are some similarities for sure, right? Like probably some of us, who have the COVID vaccine, then you would have protection against COVID, and I would have protection against COVID. Okay. But you might not have protection against hepatitis. And I do because I receive a shot for it, right? So it depends what you are exposed to throughout your lifetime. And that's why this kind of immunity is number one, adaptive, it changes, and number two, it's acquired because you're not born with it. Okay, you have to develop it by being exposed to either the real thing or to a vaccine, which is like we'll talk more about vaccines, okay? But vaccine is not necessarily the real thing, right? It's close to the real thing. It's like, educate your immune system and gets it ready for the actual infection, basically. All right. So innate and adaptive. Here is a list of innate immunity mechanisms. These are ways to protect you from infection, barriers to entry, inflammation, so on and so forth. We'll learn about each one of this today. I think that's probably all the time we have. We'll see. Adaptive immunity is going to involve production of specialized cells, the T cells and the B cells, and production of antibodies. Let's start with innate immune system. Remember, these are things that we are born with. As soon as the baby comes out, it's gonna have all these innate immunity ready to go. Okay? The baby is really protected in the mother's womb. It's not exposed to any pathogens or anything like that. But once it comes out, the air is full of pathogens, right? The blanket that you wrap them in, okay, it's probably going to have some pathogens, right? And again, like, not all of them are going to cause a person's very, very ill, but but they have the potential to, right? Let's start with physical barriers. This is very straightforward. The best protection is to keep things out of your body. Right? If you don't let them come in, then they cannot make you sick. Right? So that's why we wash our hands. Right? So when we eat, we're not going to introduce unnecessary pathogens into the body. Right? We try to disinfect the surfaces we touch, so on and so forth. But on our body, we have physical barriers. Right? Intact skin. Skin is a pretty effective physical barrier to prevent infection. Okay? If you have a cut in the skin, then it gives opportunity for things to go in. Okay? Sometimes in the winter, people get really, really dry skin if they don't put lotion on, and then the around the fingernails, the skin start to crack, and then the bacteria can start going in, and it causes cellulitis. Okay, like you will have these infections in the fingers, in the around the nails, right? And you have to put antibiotic ointments on them, right? So intact skin is important. People who suffer from burns, right? If it's very severe burn, they actually have to be kept in a sterile environment, right? Because if the skin is damaged for a bulk of the body, then things in the air can just land directly on you and cause infection, right? So very important to have skin that is intact. But you cannot have skin over the entire body. Like for example, like inside your mouth, you can have like these kind of skin. Your eyes, sure, you have the eyelids, but when you open them, you can't have skin on the eyeball because then you won't be able to see, right? So for places where you don't have skin, you have mucous membrane instead. So it's not just the when we think about exposed surfaces, people often think about what you can see on the outside, like oh, this is exposed, my face is exposed, like my mouth is exposed. But those are not it, right? Your entire digestive tract is actually exposed to the outside. If you think about it, right? Like yeah, this is the entrance for food, and then it comes out on the other end. The whole thing right inside where my finger is, that is also exposed surface. You can't see it because it's inside you, but even though it's inside you, it's still kind of outside, right? Because food that you put in is going to pass through it. So those locations, you need to protect it with mucous membrane. Right? Same thing with respiratory tract, reproductive tracts, and urinary tracts. These are all exposed surfaces. Some of these surfaces will have ciliated cells. They keep on, doing the sweeping motion, and the sweeping motion achieves two things. Number one, it prevents things from sticking to them. Right? If the surface keeps on moving, it makes it harder for the bacteria to grab a hole and stay there. And it also helps remove a mucus, which might trap some of the particles that we don't want, uh, and move it up to the throat so you can spit it out or swallow it. Okay, so these are physical barriers. But physical barriers alone are not enough. Oh, sorry, I went ahead of myself. Another example of physical barriers are these things called normal floras. Normal floras are microorganisms that live on us or in our body. You have a lot of uh, beneficial bacteria that's living in you, in your guts. They help you break down food sometimes. They help you make vitamins. We give them a place to live, and then in turns they help us have a healthy, gut community. Okay, and how do you get more normal floras? Like if sometimes you have a like upset stomach and then you get food poisoning, diarrhea episodes, what do people take to replenish the um, the gut normal flora? You guys know what I'm talking about? How do you put more good bacteria in your system? That's my question. Yeah, that's right. Probiotics. Okay, probiotics. Probiotics are these healthy, beneficial bacteria that will help populate your intestinal tract, and by taking up so much space in your intestinal tract, it prevents the harmful ones from taking up residence. Okay, so it's filled with good bacteria. When a bad one comes along and is trying to attach itself onto it, there is basically no room for it, right? So that's how normal flora protects you. Here are some normal floras that are commonly found in the body and on the body, and they have a first name and a last name. Staphylococcus epidermidis. Here is another one. Propionibacterium acne. And you can tell these things, the name, uh, kind of describes problems that they might cause. So, for example, this one can cause some kind of skin infection. This one can cause acne. And then we have something called Staphylococcus aureus. We'll come back to that in a second. But under the right condition, they might penetrate the skin and other protective barriers and do cause problem. So normally they protect you, but if the opportunity arises, they could also cause a problem. Right? So Staphylococcus aureus, or just S. aureus, is particularly concerning for healthcare because there is something called MRSA. Okay? If you've been hospitalized, they probably ask you whether you have been infected with MRSA before, and then they might do a little sampling, collect a little bit sample from you to basically test to see whether you have this thing on you. MRSA stands for methicillin resistant S. aureus. So these bacteria sometimes can evolve and pick up plasmid. We learned about plasmid last semester, right? So they could pick up plasmid from other bacteria and cause them to be resistant to even the strongest class of antibiotics. Okay, and one of the strongest class of antibiotic is called methicillin. If they're resistant to that, then it becomes very difficult, if not impossible, to treat them. Okay, so that's why they're very concerned about MRSA, because if you have it and you go into a hospital, you can spread it to other people as well. Right? Because everybody has S. aureus on them. Not all of them has resistance, But if you get exposed to a resistant one, it will turn your normal bacteria into resistance because, like I said, they could exchange plasmid, right? Okay, chemical barriers. Chemical barriers are additional protection. So, for example, we have lysozyme, right, which is a kind of antibacterial enzyme, and you have it in sweat, in tears, and in saliva, right? Because the eyes and the mouth, that's just like teeming with bacteria all the time. Yeah, your eyeballs constantly have a bacteria landing on it from the air. So every time you blink, you basically wipe it with a film of tears, and in that tears, you will have some lysozyme to keep the bacterial population under control. If you rub your eyes a lot with your hands, then you introduce even more bacteria, and sometimes it can overwhelm what the lysozyme can cope with, and you get an eye infection, right? A pink eye, right? We also have stomach acid, the hydrochloric acid, which of course will kill most of the bacteria in the food that we eat. Although sometimes there are certain bacteria that are very adapted to the acidic environment, and they could hide in the stomach. We'll learn something called the H. pylori later on in the digestive system. And if they survive the trip in the stomach, then they could cause, upset stomach and diarrhea later on in the digestive tract. Okay, but for the most part, the acid in the stomach is pretty strong. It will kill most of the bad stuff. Okay, the vagina is also slightly acidic, which is important for preventing fungal growth and bacterial growth as well. Okay. Sometimes in pregnancy, they would screen for certain kind of bacteria that is growing in the vagina of the mother. And if they screen positive, uh, something called like group B strep, right? Then what happens is they have to put the mothers on antibiotics because otherwise, when the baby is delivered, it's possible for some of those bacteria to get on the baby, okay? And then it infects them during birth, right? So again, the low pH is going to help most of the time, but sometimes it's not enough. So there you go, physical and chemical barriers. That's about keeping bad things out of the body, essentially. If they do get into the body, what do we do? Well, one of the first thing that happens in the body in response to an infection is inflammation. So let's take a note on that in case you're falling asleep from listening to me talk too much. Let's write something, give you something to do, make this more engaging. So here we go. It's a short note, right? This is lecture 3, and this is about inflammation. When we discuss these immune responses, I want you to think about the purpose of each of the things, okay? So for the chemical barriers and the physical barriers, excuse me, the purpose is to keep pathogens out of our body, out of our tissue, right? What about inflammation? What is the purpose of inflammation? So let's write down. Purpose of inflammation is to recruit more WBC, white blood cells, and plasma protein to the site of infection, right? So what do the white cells do? Why do you want more of them? Obviously, the white cell is going to help clear the infection, okay? I'm just gonna use bacteria as an example, okay? It's true for other infection as well. Right? Bacteria, people tend to relate to it more easily for some reason. Right? So like if you get infected with the bacteria, then let's call some more white cells, let them come, let them eat them up, right, and clear the infection. And we're gonna get more plasma protein, right? And this will help with the healing process. Okay, healing process. And as you will see, some of these plasma protein will actually enhance the immune response, right? But let's just keep it like that for now. All right, this is going to be a tough question, okay? Like I told you this before. Let's see if you have it in your notes or if you remember. What is the name of the cell that's responsible for triggering inflammation? I'll give you a hint. It starts with the letter M. Okay. Find it in your notes. What is the name of the cell that is responsible for triggering localized inflammation? Let me know when you found it. Okay, MRSA is the bacteria. Okay, that's like the bad thing. Okay, but the cell that trigger inflammation in the body is called mast cell. Okay, so mast cells. Mast cells are granulocytes that uh, stay in tissue and they release histamine to trigger inflammation. Okay. In response to injury or infection. Okay. Like if you have a cut, you're gonna have inflammation as well. Right? How do you know you have inflammation? What are some of the characteristics of an inflammation? Think about last time you cut your finger, okay? Uh, how how does it feel? Right? There are four things. Yeah, it's gonna be swollen. Okay, and the swelling is gonna make it tender. Yeah, there's heat. It feels warm, very good. Okay, what else? Next one is very obvious. Okay, like yeah, that discoloration. But usually, like, what color is it? Are you thinking about? Like instead of like skin color, it's gonna appear a little bit red. That's right. Okay, so it's gonna be swollen. It's gonna be warm with the heat, and it's gonna be red. And finally, this last one. Okay, maybe it's too obvious. You don't think it's a characteristic. It's gonna hurt, okay? Pain. So those four things are characteristics of an inflammation. Let's find out why. Let's see what the histamine will do. Okay, so histamine. Okay. So these mast cells, they're gonna release the histamine, and the histamine has two effects on blood vessels. Okay, number one, vasodilation. Okay. Vasodilation means it will increase the diameter of blood vessel. Increase the diameter of blood vessel. So how is that help? How does that help? Well, you're trying to recruit, trying to get more white cells and more plasma protein to the site of infection, right? So by making the diameter of the blood vessels bigger, right? The effect of that is increases blood flow to the site, right? And with increased blood flow, more WBC and plasma protein can get to the site. So you make the vessel bigger, increases blood flow. Along with the blood, you will bring more white cells, WBC, and more plasma protein. However, what's the side effect of this? Side effects. Because we are bringing more blood, it's going to feel warmth, right? The heat that you guys were talking about, and redness. It's going to look red. Right? To help you understand this, like think about in like an embarrassing situation, right? Your face feels warm and it flushes, right? That's basically vasodilation going to the face, right? But that's not related to inflammation. Like I'm just trying to tell you how warmth and redness is associated with increased blood flow. Okay, so that's the vasodilation. Second effect of histamine on the blood vessel is that now that we have all these white cells coming in and all these plasma protein coming in, we want to help them get out of the blood vessels, right? Because look, over here, if we have an infection, like the bacteria are going to be like right here, right? So after the white cell gets here, we want to help them go into the tissue and find those bacteria, right? So the second function is going to be increase permeability. Okay, that's okay, Michael. Thanks for letting me know. Okay, permeability. Okay, so this allows WBC and plasma protein to leave the blood vessel and enter tissue easily. But there is a side effects associated with this. Because now the blood vessels are even more leaky than they normally would be. And sure, we have the white cells going out and the plasma protein going out, but if it's more leaky, then fluid is going to go out as well, right? And if we have more fluid going in, more than the lymphatic vessel can drain them, and we're going to have some swelling. Okay, and the swelling is going to be pressing on nerve endings in the tissue, and that's going to create pain. Okay, so these four things, these are called characteristics of inflammation. Okay, these are all marks of inflammation. And the kind of pain that's associated with inflammation is very distinct, okay? People usually call it like a throbbing pain. You feel like pulsating. Okay? Then you have an inflammation. So the purpose is not to make it red and make it all swollen and painful. These are secondary effects. These are side effects, right, as a result of the action of histamine. Okay. So histamine can cause localized inflammation. Localized means it only happens in one part of the body. But for allergies and stuff like that, histamine can cause systemic type of problems, and that's when you need to take like antihistamine, right? But there you have it. That is inflammation. Inflammation. Okay, let's go back to the PowerPoint slides here for a second. These little black circles, you just have to believe me, they are mast cells. If you zoom in on them, they look like this. And you see they have a lot of these granules. What's in those granules? You've guessed it, histamine. Okay? So when they release the histamine like that, these histamine will immediately affect the blood vessels that are nearby, making them bigger to increase blood flow, making them leakier for things to come out. Okay? So that's what they do. Mast cells. Any question? Great. We are talking about innate immunity, okay? Mechanisms that you're born with to protect you from infection. All right. Oh, there's a question. Are mast cells a type of WBC? Yes. Let me refresh your memory a little bit over here. We did it before, right? Like white blood cells, we have granulocytes and agranulocytes. Mast cells is right here. Okay. Related to mast cells would be basophils. Basophils also cause inflammation, but it's more like for the whole body as opposed to like localized. All right. That was January 15th notes. Okay, back to here. We're talking about innate immunity, right? Things that you're born with to fight infections and pathogens and whatnot. So let's just do a few easy poll questions. Go ahead. All right. Not a lot of answers coming in, but physical barriers, that would be ciliated cell. Okay. If it involves some kind of chemical, some kind of secretion, then it would be, chemical barrier, right? So ciliated cells are in the airway and the reproductive tracts for females, and that would help clear and prevent infection. How about this one? Okay. I suppose that's all. The answer is localized swelling. Okay, localized swelling. Okay, histamine is going to increase permeability, and because of all the stuff that are leaking out, it's going to lead to swelling. Okay, B is not correct because the these things are switched. Okay, white cells enters into the tissue from the bloodstream. Okay, it's the other way around. It doesn't go from tissue to blood. The white cells come from the blood, right? And it's going to go into the tissue. So that's why B is not correct. All right. We have the barriers to keep them in. If they fail, then it comes in. And then we have the inflammation. The inflammation will bring more white cells to the site of infection. And then what do we do next? Okay. The next innate mechanism is called phagocytosis. Okay, phagocytosis we learned last semester. It's cell eating. So not all white cells can do this. Some can. And of the cells that we've learned, neutrophils and macrophages are white cells that can do phagocytosis. Right? We call them phagocytes. Site is cell. So a cell that can do phagocytosis is a phagocyte. Okay. So what do they do? Well, they basically are going to eat um, things that are not supposed to be in the body. It doesn't even have to be like bacteria necessarily. It could eat up old or dying cells, right? There are often signals on these cells that would trigger them to eat them. And these signals that are on the surface of the cell that would trigger the phagocytosis, we call them PAMPs. Okay, PAMPs. I'll write down what PAMPs stands for you. Pathogen, pathogen associated molecular pattern. Right, that's what PAMs are. Molecular patterns, it's a group of molecules, kind of like a barcode, if you would, that is on the surface of a cell. And if these barcode register as a pathogen, then the phagocytes will go eat them. Okay. In fact, the phagocytes is gonna check the cells that comes into contact with them, and if it doesn't have the if it has your own antigens, then it's gonna leave it alone. Right? If it has these PAMs on it, then it's gonna eat them. Okay. Sometimes the existing phagocytes in our system is not enough to fight off the numbers of bacteria that is coming in. Okay. And so they need to call for backup. They need to call for reinforcement. And to do that, they release some kind of chemical signals called the colony stimulating factor. These colony stimulating factor will travel through the bloodstream to the bone marrows and it will increase the production of white cells. I told you already, bone marrow is where you will produce all the different kinds of blood cells, white, red, platelets. But in this particular instance, when you are trying to fight off bacteria coming in, then the colony stimulating factor is going to ramp up the white cell production. Okay. Just like when your body needs oxygen, the EPO that we talk about, EPO, it's going to ramp up red cell production, right? So depending on what your body needs, you can, tweak the bone marrow production to increase a particular kind of cells that you need. Okay, so that's phagocytosis. The blue here is uh, uh, a macrophage, and then this here is a cell that contains the PAMs. Okay, so on the surface they have these barcodes, these molecular patterns, which the white cells recognizes as enemy, basically. As the infection is being overcome, right? So in the best case scenario, all these extra white cells coming in, it's gonna eat up everything. Right? They could eat a lot. Okay, the phagocyte they could eat up to hundreds of bacteria before it dies. Okay, some of the phagocytes will die, right? From fighting all these bacteria, and so the dead phagocytes, along with some dead tissue cells, dead bacteria, and even living ones possibly together they form the pus. Right? Pus is, I'm sure you know what it is. Is that the, whitish stuff, sometimes yellowish fluid that's sometimes forming in a wound, right? Like in the pimple, for example, that's the pus, and it contains all these things, right? And because it could potentially contain live bacteria in it, pus is an infectious fluid, okay? So you shouldn't really uh, be, touching uh, us without a glove, okay? Basically. I mean, your own is okay, but like not others. That's what I'm trying to say, right? Like it can spread diseases. So the presence of pus indicates that the body is destroying dead tissue cells and is trying to overcome an infection. So pus is good, okay? But you don't want too much of it building up. It's possible that there's an excess of pus building up sometimes in cavities in the body. Then you have to go to the hospital and drain it. But generally, a little bit of pus just tells you that you're fighting an infection and it's going to get better. Now, this is a very good picture to spend some time to look at and study it. It summarizes everything that we've learned up to this point. So let me go through it with you. So first, there is some kind of injury coming in. This is a splinter, for example, going into your skin. Okay, punctures the skin and basically breached the physical barrier, right? And along with the splinter, you introduce some bacteria into your tissue. Okay. So right away, the mast cell that has been sitting there is going to release the histamine uh, in response to the injury and the presence of these pathogens. What do the histamine do? The histamine will increase the blood flow by making the blood vessel bigger. Right? So more white cells is going to be able to come, and some plasma protein will be able to come as well, and it makes it more leaky. Okay, so here it says increased permeability of capillary causes local accumulation, buildup of tissue fluid, and then there's the swelling. The swelling will stimulate the nerve endings, which is what this yellow thing is, and it causes pain. Okay? So, of course, because of the injury, we want to initiate a blood clot, and that's why we need the plasma protein, right? Okay, earlier in lecture two, we talked about the fibrinogen, the prothrombin. All that plasma protein needs to come and help initiate the blood clot to basically seal the gap, right? We don't want more things coming into the blood. At the same time, we have some macrophage already in the tissue, right, and neutrophils perhaps, and they are going to start eating up the pathogens, eating up the bacteria. How do they know the bacteria are bad? Well, they check the cells and they detected the PAMs on them, right? The pathogen associated molecular patterns. These barcodes that tells the pathogen, that tells the macrophage, these are bacteria. So they will eat them, but more often than not, the macrophage cannot eat all the bacteria that are coming in. We're literally talking about like thousands and thousands of them, right? And so the macrophage will call for backup. And the way it does that is to release these chemical signals called colony stimulating factors. They will go to the bone marrow and it will crank up the production of more white cells, particularly the ones that can do phagocytosis, such as neutrophils and macrophage. All right. Now, here are some monocytes I know it's a lot of information, okay? Before I told you, under white cells, the agranulocytes, the monocytes are inactive, and once they are exposed to bacteria, for example, then they can change into the active macrophage. Okay. So over here, these are like the new guys, the new recruits, fresh out from the bone marrow, fresh out from the academy. They don't know how to fight yet. But once they see the bacteria, they will transform into the macrophage, and then they will start eating things. Okay. That's the story so far. And if all goes well, then your infection is going to be cleared and everything is right as rain. Questions? No questions. All right. Now you just have to remember these things, right, for the test. I don't know when the test is, probably sometime later in a few weeks. Okay, we got another group of cells that are going to be able to help us, with our infection, and these are natural killer cells. Okay, natural killer cells, which stands for sorry, the short form is NK cells. Okay, and again, we actually wrote this down before, right? Right here, right? It's a type of lymphocytes, right? T cells, B cells, and NK cells, natural killer cells. What do they do? They kill virus infected cells, okay, as well as tumor cells. Just to help you refresh your memory a little bit, uh, this is a host cell. I know it says bacteria cell here, but it's same. It could be your cells, okay? And then we have some kind of virus infecting them. The virus will inject viral particles into viral DNA into it, and then it will force the cell to make viral particles, and then the virus will assemble and then will burst from the cell, and then they will go infect others, right? We learned this last semester, right? So this is what happens. When a cell is infected with a virus, when your cells are infected with a virus, sometimes they would put distress signal on the surface, okay? It's basically telling the natural killer cells, hey, I'm infected. All right. The viruses are growing in me. They will burst out any moment. Okay. Please kill me before the virus comes out. Because once the virus comes out, they will, spread and infect other cells, right? So it's like a zombie movie, right? If one of the characters in the zombie movie gets bitten, they might say, okay, you gotta kill me before I turn into a zombie. Because as soon as they turn into a zombie, they will bite other people, and other people will turn as well, right? And that's the job of NK cells. They're gonna do the killing. If they detect a cell has been infected, they will destroy them before the virus pops out. They are also reasonably okay at killing tumor cells. Okay. In fact, tumor cells, which are also we learned last semester, they are cells that has mutations and they could grow into a tumor, right? Which might or might not be cancerous. But these tumor cells regularly form in the body. Okay. I heard a podcast recently, and then the researcher in the podcast was saying, every day there are like 4 or 5 tumor cells that are being formed, and most of the time we don't notice, and that's because the NK cells are doing their job. Okay, the NK cells are clearing them out before they full-fledged become the big tumor. Right? But sometimes they fail, and that's how people get either benign tumors or malignant tumors in some cases. So here the orangey guys, they are the NK cells. You just have to believe me. And then the red one would be some kind of tumor cells or virally infected cells. Okay. Next, we have the complement proteins. We're almost there, guys. Okay, stay with me. Complement proteins. Complement proteins are plasma proteins that are primarily made by the liver. Liver is on the right side. of your body under the diaphragm. And a lot of the plasma proteins are made in the liver. We talked about albumin already, right? The clotting proteins, the fibrinogen, the prothrombin, they are also made in the liver. And now we have a bunch of these things called complement proteins, which are also made in the liver. Complement proteins are in your bloodstream. They're circulating. It's part of your plasma, and they are mostly turned off unless you are fighting some kind of pathogens. Okay, so most of them are inactive. You need to turn them on when there is an infection. And to turn them on, you have to do a series of chemical reactions. It's really complicated, okay? Like that's what this picture is showing. So for example, there's a classical pathway. You will turn on C1Q, which then turn on C1R, and so on and so forth. And after you activate all these, then it will turn on C3, and so on and so forth. The point, it is like a domino. Okay, like you put the dominoes on the table, you push one, and then it goes all the way. One thing leads to the next. Ultimately, what is the point? What is the point of complement proteins, right? The main thing to remember about complement proteins is that it helps synergize other immune defenses. Okay. Synergize means to make them better. Okay. They work with each other and together they achieve something greater that individually you cannot do. Okay. That's what synergize means. Okay. It makes it better, even stronger. So I'm going to give you three examples of how complement proteins help with the infection. Now, number one, complement proteins, once you activate them, is going to help recruit more white blood cells. Okay? And you're like, wait a minute, isn't that the function of inflammation? Right? The purpose is to recruit more white blood cells. And you are right. Right? And that brings me back to the point I was trying to make. The complement protein just makes the thing better. It synergizes it. Okay. Inflammation wants to recruit white cells, no problem. I'm gonna help make this even better, right? We're gonna help recruit even more white cells. So how does it do it? The complement protein basically leaves behind a chemical trail for the protein for the white cells to follow. Okay. Imagine if I am a white cells that just came out of the bone marrow. Okay. I heard the backup call from the white cells. Okay. So, I came out of the bone marrow, but the body is so big. How do I know where to go? Right. That's where the complement protein is going to be important. It leaves behind a chemical trail, like the story Hansel and Gretel. Okay. They leave behind the breadcrumbs, right? For them to follow, right? And eventually, the white cells will be able to arrive at the site of infection. All right? If this is still confusing, I'll give you another analogy. Right? If you want to look for the cafeteria in a building, sometimes you can follow the smell, right? Okay, without the smell, you don't know where to go. Okay, but if you can smell the food, okay, the fried chicken or whatever, then you kind of know which direction to go. That's what the complement protein does. It leaves a trail, chemical trail, for the white cells to go. Okay? That's number one. Number two, the complement protein is going to help coat the target. So the white cell that came out from the bone marrow followed the chemical trail to the site of infection. It squeezed itself out of the blood vessel and now it's in the tissue. Okay? I am trying to eat this bacteria, right? That's infecting the body. But in order for me to do that, I have to check. I have to check the cell that comes into contact with me, right? Okay, I have to check to see if they have that barcode, right? The PAMs, right? And that's time consuming, right? And so what complement protein does is it can flag the target cells, it can flag the bacteria and highlight them and put a please eat sign on them. So when the white cells come out of the blood vessel, it can see the bacteria right away. It's like, oh, that's the thing I gotta eat, and it's gonna make a beeline to it and do phagocytosis on it. It's like this game, right? You might have played this when you were younger, or like my kids love to play this. It's called Finding Waldo, right? In this picture, you have to find Waldo, this guy. Okay, so like you can take a moment and try to find it. When you're looking for Waldo, it's like the white cell looking for which cell to eat. Right? So like I got to check each cell. Like, is it this one that I gotta eat? Is it this one? Is it this one? All right, it's gonna take some time. Eventually, you're gonna find it. Okay. And what complement does is this. Complement is gonna put a big red circle around the thing you're supposed to eat. Okay. So when the bacteria, when the white cell comes out, immediately it can identify the target. It's gonna walk over there, eat it up, and then help clear the infection faster. Remember, complement is to synergize other processes. It makes them better and easier, right? And by flagging the target, it allows phagocytosis to happen faster. Okay? So far, the two things I told you about complements are more about like enhancing other immune response, right? But complement can actually do a little bit of attacking as well. And that's the third function I want to talk about, right? These complement proteins, once you activate them, some of the pathways is going to allow them to join together to form what's called membrane attack complexes or MAC. Right? These membrane attack complexes is exactly what it sounds like. They are going to uh, put themselves into the bacteria cell wall and punch holes in them. Once the bacteria is full of holes, then water is going to go in and it causes them to burst and then it kills them. Okay. So in addition to recruiting more white cells, helping them with the phagocytosis, complements can do a little bit of attacking on its own by forming these membrane attack complexes. Okay. So these are just some of the things complements can do. There are a lot more that we're not going to go into. Quick recap. Physical chemical barriers to keep them out, inflammation to recruit the white cells if they're in, and then the white cells will do phagocytosis to try to clear it out. NK cells will help a little bit in the case of viral infection. And to make these things even better, we have the complement proteins to enhance them, to synergize them. All right? I hope you're with me. Whole time. Yeah, I'm not getting a lot of responses. Can you guys choose something, please? All right, I think that's all I'm gonna get from you guys. The correct answer is pathogens. Okay, pathogens. Yeah, PAM is pathogen associated molecular patterns. So of course you're gonna find it on the pathogens. Okay, it's the barcode on the pathogens. It's not on the phagocytes. Okay, a lot of people chose phagocyte. The phagocyte is the white cells that will recognize the PAMs and then they will eat it. Okay? Anything with a PAM on it, the phagocyte will eat. Okay, let's try this next question. And I'm looking for a student, okay? Sarah El Sayed, if you're here, please send me a message, okay? Sarah El Sayed, please send me a message. I need to find you. Thank you. Okay. Higher than normal chance of developing malignant tumors, that would be NK cells. If you don't have the NK cells, you cannot clear the tumor cells, and they might grow, right? So that's the answer. Last one. Should be easy, this one. Okay. If you have been listening to the lecture, then you will know the membrane attack complexes are formed by complement proteins. Complement protein. All right, the last mechanism that we're going to look at for innate immunity is fever. A fever is a temporary rise in temperature above the normal. If use Celsius, then it's 37 degrees Celsius, which is 98.6 degree Fahrenheit. The fever can go pretty high, right? My daughter actually just had a fever over the weekend and it went as high as 40, right? So that's a sign that your body is trying to clear the infection. And your body secretes these chemicals called prostaglandins, which are pyrogens, okay? They crank up the body temperature. There is a part of the brain, which we'll learn in the brain unit, so don't worry too much about it for now. It's called the hypothalamus. Right? Of all the things it does, it helps regulate body temperature. So the prostaglandin will turn up the temperature, tell the hypothalamus to turn up the body temperature. What's the purpose of fever? Fever will help enhance the immune defense by speeding everything up, basically, okay, by pressing like the fast forward button in a movie. Okay, it helps increases enzymatic activity. Okay, it helps speed up metabolism, like all the chemical reactions that are happening. And these chemical reactions might help with wound healings and create activating complements, for example, and also increasing the rate of tissue repairing and phagocytosis activity. Okay, of course, not included on this list. Fever will also reduce pathogen activity. Okay, the heat is going to help slow down the bacterial growth, and it keeps them at bay while you're trying to clear them out. Right, so that's where the hypothalamus is. It's very deep in the brain. Okay, it's very small, tiny area. So that's fever. Right? So like unless the fever is really, really high and there are other symptoms, sometimes maybe it's better to just let it run its course because it is helping you to clear the infection. But to make the kids feel more comfortable, like I had to give her some Tylenol and stuff like that. So that is it for innate immunity. Okay, I think that's enough for today. Lots of things for you to work on your own. The study guide, and just go through, organize the notes a little bit. Um, I don't think there's anything urgent coming up. Let me quickly check the calendar, and then we're gonna dismiss the class. Let me just go back to here. I guess you should start working on your labs actually because you've done two and a half lesson already, so you should be able to start working on the lab. But in terms of um, things that are coming up, let me see. Okay, the lab quiz is due on February 5th, okay, which is next week. Yeah, so we have time. And then the test is not until February 7th. Okay, so just keep those in mind. But if you want to start on the lab, what you can do is you can scroll down and find the lab, which is right here, lab one. And you have to download the PowerPoint and work on the questions in the PowerPoint. Okay? The models are discussed in the lab videos, so watch it and then do the questions. You don't have to hand them in. Okay? Doing these questions will help you prepare for the lab quiz. The lab quiz will be 3.3% of your final grade. It's weird, okay? We have three labs. All the labs are worth 10% in total. So one lab is 3.3%, okay, roughly. So that is that. By the way, are you guys able to see your grades for the quiz? Um, like I said, it's a new system, so I'm not. Yeah, instantly. Okay, yeah, that's okay. That's something I have to change. Next time it won't be instantly, like it would until everyone else finish first. Okay, but I think it's pretty good. The class average is 79. Okay, so keep up the good work and enjoy the rest of your week, guys. Okay, take care. Bye for now. And Sarah, if you can hear me, okay, I can see you on the list. Please send me a message and stay behind. I need to talk to you. Okay, otherwise. Goodbye, everyone.
Goodbye, sir.
Hi, Sarah. Can you hear me? Or no, you're not there. No, not there.